DNA, RNA, and protein synthesis. So far, we've mentioned that DNA is within the nucleus, specifically in chromatin fibers that are made of DNA and protein. We've also talked about how the nucleus is the control center and that DNA is an important part of that. Now we're going to look more into that structure, the differences between DNA and RNA, and how that's used to make proteins. Models of DNA show a double helix. Although Watson and Crick, along with Maurice Wilkins, won the Nobel Prize for the discovery of this double helix in 1962, there are several other scientists that were really instrumental in discovering this, one of those being Rosalind Franklin, who, using X-ray diffraction, was able to show that DNA is a double helix. The structure of DNA. James Watson and Francis Crick had several puzzle pieces as they determined the structure of DNA in 1953. They knew that DNA was a polymer of nucleotides and that each nucleotide is a complex of 3 subunits. We have a phosphoric acid or phosphate, a pentose sugar, for DNA it's deoxyribose, and a nitrogen-containing base. They also knew from Erwin Chargaff's work that the amount of adenine always equals the amount of thymine, and the amount of guanine equals the amount of cytosine. These are the nitrogen-containing bases: adenine, thymine, cytosine, and guanine. Back in Chapter 2, we talked about how nucleic acid, which is the polymer, is made of nucleotide monomers. This is an example of a nucleotide. We have a phosphate, a pyrimidine base, and so here is one section, one nucleotide. It joins together with another nucleotide, again with a phosphate and a purine base. So pyrimidines join with purines. We'll look at the differences between those momentarily. This unit here represents one rung on the DNA ladder. We just mentioned that there are 4 possible bases in DNA. 2 are purines, adenine and guanine, A and G, and 2 pyrimidines, thymine and cytosine, T and C. DNA is a polynucleotide strand with a backbone of alternating phosphate and sugar groups. These 2 strands make up a double helix. It looks like a twisted ladder. The strands are held together by hydrogen bonding. Earlier in Chapter 2, we talked about how hydrogen bonds are weak individually but strong collectively. We'll see how this is really useful for the structure of DNA, since that double helix is unzipped to access the genetic information contained within that DNA. There are complementary base pairing within DNA. Adenine always pairs with thymine, and guanine always pairs with cytosine. The 2 strands are anti-parallel, that is, they're oriented in opposite directions. The sugars are oriented differently. There's a 5' free end on one sugar and a 3' free end on the other sugar. I'll show you. This is what they mean. Whenever they are numbering the carbon atoms within this phosphate, they number them 1, 2, 3, 4, and 5. This is what they're referring to as the 5' end or the 3' end, and when things are anti-parallel, going just in opposite directions. When DNA is replicated, it's the process of copying one DNA double helix into 2 identical double helices. Since adenine only bonds with thymine and cytosine only bonds with guanine, when the strand is unzipped, we have a template to make new strands of DNA. This is how we can get accurate replication. Here's an overview of how it works. Here's your original strand, and as these hydrogen bonds are broken in between the bases, then other nucleotides can come and match up to the appropriate base on the other end. The daughter DNA double helix, this is said to be semi-conservative since we have half of the old strand and half is composed of a new strand. During transcription, a gene, which for now we'll just refer to as a segment of DNA, it serves as a template to produce an RNA molecule. Earlier we said that RNA was a single strand and that instead of having thymine as a base, we have uracil. The historical definition of a gene was a nucleic acid that coded for sequences of amino acids in a protein. The proposed new definition is similar. A gene is a genomic sequence, either DNA or RNA, directly encoding functional products, either RNA or protein. So a slight change there, but as we look at this concept of transcription and translation and how we're getting information from that DNA strand, this will make more sense. The genetic code. So how are we getting information from DNA? Within that nucleotide sequence, there's coded information. There's a triplet code. Each 3 nucleotide unit of an mRNA, that's messenger RNA, we'll look at that more momentarily, is called a codon. There are 64 different mRNA codons. 61 of those code for particular amino acids. And if we're recalling also from chapter 2, amino acids are those monomers that make up protein polymers. And we also recall that proteins have a lot of different forms and functions. Within this, there's a redundant code. Some amino acids have numerous code words, which provides for some protection against mutation if you have several different ways to get the same protein. The other 3 of those codons are stop codons that signal termination. This genetic code is almost universal in all living organisms, which makes sense since we're all basically made up of the same stuff. During transcription, we take that code of DNA, it's unwound and read or transcribed into a messenger RNA molecule. So this is representing that double helix of DNA. If we have a segment of this that we're reading as a gene, then mRNA, messenger RNA, can be read and created. This is an mRNA transcript. After transcription, we move to translation. Translation is the second process by which gene expression leads to protein synthesis. All of this is looking at how do you read a section of DNA and get that final product of a new protein. So first we have DNA replication, then transcription, getting a section of messenger RNA. Now we have translation. Translation requires several enzymes and different types of RNA molecules. We have mRNA, which is messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. Translation requires 3 steps. During translation, codons of that mRNA, and that's a triplet code within those codons, base pair with anticodons in the transfer RNA carrying specific amino acids. The order of codons determines the order of transfer RNA entering the ribosome and the sequence of amino acids in a polypeptide. All this is saying is that the code within that messenger RNA is going to determine the sequence of amino acids that are brought. Earlier, we talked about how a ribosome, which is where proteins are made, has 2 subunits, a small subunit and a large subunit. What happens is a messenger RNA slips between these, and this codon is read. This is represented by that tRNA binding site here. So as mRNA is read, that transfer RNA brings an amino acid and starts building on to the string of amino acids that becomes a polypeptide. This eventually will be processed and folded into an appropriate protein. Reviewing gene expression, first, within the nucleus, we have DNA that serves as a template for messenger RNA. Second, messenger RNA is processed before leaving the nucleus, comes out through that nuclear pore within the nuclear envelope. Then, messenger RNA moves into the cytoplasm of the cell and becomes associated with ribosomes. And here, showing those two subunits, the large and small of a ribosome. Next, transfer RNA with anticodons carry amino acids to that mRNA as it reads that triplet code. Next, the anticodon-codon complementary base pairing occurs. Finally, polypeptide synthesis takes place one amino acid at a time. And this is how we build that polypeptide.